Have you ever wondered how scientists isolate antibodies, those tiny disease-fighting proteins? It's like finding a needle in a haystack. But luckily, we have clever tools like protein A chromatography resins to help us. Imagine these resins as tiny magnetic beads. Each bead is specially designed to attract and capture antibodies from a complex mixture, leaving behind all the unwanted stuff. This process, called affinity chromatography, is vital in producing pure antibodies for research and medicine. These purified antibodies are then used in a wide range of applications, from diagnosing diseases to treating cancers and autoimmune disorders. Without protein A resins, producing these life-saving therapies would be much more difficult and expensive. So how are these tiny but mighty helpers made? Let's take a closer look at the fascinating step-by-step process. The key ingredient in protein A chromatography resins is, unsurprisingly, protein A. This remarkable protein, naturally found in the cell wall of certain bacteria, has a unique ability. It binds strongly to antibodies. To produce large quantities of protein A for resin manufacturing, scientists use a clever trick. They insert the gene responsible for protein A production into a host organism, like the bacterium E. coli. This engineered E. coli then becomes a mini factory churning out protein A. After growing the bacteria in large vats, the next step is to extract and purify the precious protein A, ensuring it's free from any impurities and ready for the next stage of resin production. Now that we have our star player, protein A, we need a suitable stage for it to work its magic. This is where the base matrix comes in. Picture the base matrix as tiny, porous beads made from a material like agarose or polystyrene. These beads act as the scaffold for protein A attachment, providing a large surface area for antibody capture. Choosing the right base matrix is crucial. It needs to be chemically inert, meaning it won't react with the antibodies or interfere with the purification process. The matrix also needs to be mechanically stable, able to withstand the pressures and flow rates involved in chromatography. Section 4. Ready, set, bind, activating the matrix. Before we can attach protein A to the base matrix, we need to activate it. Imagine this step as creating tiny hooks on the surface of the beads, ready to grab onto protein A. Activation usually involves chemically treating the matrix with a reagent, creating reactive groups that can form stable bonds with protein A. The choice of activation method depends on the type of base matrix and the desired binding chemistry. The goal is to create a strong and stable linkage between protein A and the matrix, ensuring efficient antibody capture during chromatography. Section 5. Joining forces, coupling protein A to the matrix. With the activated matrix ready, it's time to introduce protein A. This step, called coupling, involves incubating the activated matrix with a solution containing purified protein A. The reactive groups on the matrix readily bond with specific amino acid residues on protein A, forming a stable and lasting attachment. The density of protein A on the matrix, known as ligand density, is crucial. Higher ligand density generally means greater antibody binding capacity, but it's a balancing act. Overcrowding the matrix with protein A can hinder antibody access and reduce purification efficiency. Section 6. Ensuring quality and performance, from blocking to packaging. After coupling, any unreacted sites on the matrix must be blocked to prevent non-specific binding during chromatography. This involves incubating the matrix with a blocking agent that caps any remaining reactive groups. The resin then undergoes extensive washing to remove any unbound protein A, blocking agents or other residues. Rigorous quality control tests are performed at each stage of the process. These tests ensure the resin's performance, including its antibody binding capacity, purity, and stability. Finally, the resin is carefully packaged and stored under controlled conditions to preserve its activity and ensure a long shelf life. Section 7. The future is pure. The importance of protein A resins. Protein A chromatography resins are essential tools in biopharmaceutical manufacturing, enabling the production of high-purity antibodies for a wide range of applications. From diagnosing diseases to developing novel therapies, these tiny beads play a big role in advancing healthcare. As research continues and new antibody-based treatments emerge, the demand for high-quality protein A resins will only increase. Understanding the intricate process behind their manufacture highlights the precision and care required to create these powerful tools for scientific discovery and improving human health.